been extremely lucky in some of the things I've been doing. But it couldn't be more fitting to talk to you about what I'm going to discuss today when we're talking about a STEM day, and that's because we're going to talk a little bit about stem cells. So I think it's a little bit fitting, and we're going to try to rope in how stem cells and chemistry kind of overlap and figure out how these two fields actually interact with each other. But my main purpose today, the reason you guys are here, right, is to convince you that a career and a major in one of these STEM fields is worth the while. And so actually what I'm supposed to do today is to turn you into somebody like me, I think. Okay? And so you can see me standing up here, and I feel a little bit overdressed, and so I'm going to take this off. And now I've really moved forward here quickly. And turn you into somebody that looks like this. That's what I'm supposed to do today. Do you guys know who this is? Beaker. Yeah, this is the guy that actually got me involved in science at a very, very early age. Okay? I love this guy. This guy's crazy. This guy's nuts. Okay? And so this is my job today, guys, is to convince you through some of the things that I've been lucky enough involved with through my time at SFA, through my time at A&M and whatnot, that doing these kind of things is worthwhile. And so the next thing I want to do is actually to tell you why I am here. You guys actually, you got shafted a little bit, I think, okay? You're supposed to have an astronaut today. And you got me, this boring chemist, up here in front of him talking to you about chemistry and stem cells, but you're supposed to have an astronaut. So, you know, I got to live up to this expectation and try to beat an astronaut, a guy that's been in space, a guy that's actually seen the world from a very different perspective, and convince you that what I'm doing is actually just as cool. But I'm actually going to think at the end of the day, I'm going to show you that it's actually even better. Okay? And we're going to see some really, really cool things today. But the reason I think SFA got me back here today and try to engage you guys in this entire arena of stem cells and chemistry is that my perspective is very different. As Dr. Frantz told you, I was lucky enough to work for Merck Pharmaceuticals, an incredible career. I actually worked on a couple of drugs that are out there on the market. One of them is Januvia. It's a type 2 diabetes drug that I was involved with. Very lucky to work on something like that. This is a drug that's taken by millions and millions of people across the entire world, and 2 people in my family actually take Januvia. And this is something I got to work on, and it all started right here at SFA. Okay? So I'm going to walk you through some of the things I'm doing now at UT San Antonio, okay, based on some of the experiences I've had in the pharmaceutical industry and now back in academia. But the reason I'm here is because I love to be here. I love to come back to SFA every single chance I get. Okay, and hopefully that will show through today as we go through some of the things that I've been doing in San Antonio. But before I do this, it looks like my presentation's nervous. It's shaking a little bit, doesn't it? What is a stem cell? What is a stem cell? I'm sure you guys have probably heard a stem cell. There's a lot of popularity in the media about stem cells. But what I want to do is get us all on the same page, okay, and at least be able to define what a stem cell is. And there are 2 basic types. One is the embryonic stem cell. These are the stem cells that we probably most think about when you hear about a stem cell. And we'll walk through the specifics about an embryonic stem cell. And then there's this new kind of paradigm that are called adult stem cells, or some people refer to these as somatic stem cells. Okay? There are differences between embryonic stem cells and adult stem cells, but the basic premise of these cells is that they are undifferentiated. In other words, they haven't decided whether they're going to be a skin cell, or a bone cell, or a blood cell. They still have this capacity to go into these different avenues and really have a great capacity to generate new tissues, okay? And a lot of these cells, especially these embryonic stem cells, they are immortal. In other words, you can culture these things indefinitely, okay? They will live basically forever, okay, until they decide to differentiate into some specialized cell, okay? So there are 3 basic types of stem cells and their ability to differentiate. And these are the 3 basic definitions that I want you guys to kind of appreciate today. The first one is totipotent. Okay? These are the cells that are at the very early stages, like within the first couple of days after fertilization, those cells that are there, these cells have the capability to give you any type of tissue. And in fact, you can take one of those cells out of that initial growing embryo and use that cell to generate an entire organism. Okay? These are master cells. These cells can do anything and generate any tissue and also generate an entire organism. Okay? These are the most powerful cells that are out there. The second set is pluripotent stem cells. Okay? They can give rise to almost every single tissue type. They can grow into cells. They can grow into hair. They can grow into bone. Okay? The only thing they can't do is they cannot give you an entire new organism. Okay? And then finally, what you have is multipotent cells. In other words, they can differentiate into several different types of tissue types, but not everything. And so maybe they're committed to a certain pathway, maybe a certain type of muscle tissue, heart muscle, okay? Or maybe a certain type of skin. But they still can differentiate into various types of tissues. Okay? And all this potential of stem cells is what everybody in the science, in this field, is very excited about, because the ability to use this power of stem cells to regenerate potential tissues, to regenerate potential organs, to solve some of our huge, huge problems and very devastating disease, including cancer and Alzheimer's. The potential of these stem cells is phenomenal, guys. It's just absolutely phenomenal. Okay? But there's a problem. There's a huge problem with stem cells, and we're going to walk you through that problem today and give you an idea of where we stand as far as the field is concerned. Okay? So as I said, the totipotent stem cells are the ones that are the very first cells within the first couple of days, okay, that the embryo is formed. These are the totipotent cells. The pluripotent cells is after like day 4 or day 5. These are the cells that have the capability, these embryonic stem cells, capability to differentiate into anything that you want, basically. And we can control this now. It's really, really cool. And I have some movies to show you guys how this happens. And then these adult stem cells are the ones that actually are committed to a certain type of lineage. Every single one of you, regardless of your age in this room, right now, have adult stem cells. Okay? We all have adult stem cells. Okay? That's how you keep regenerating your hair. That's how you heal yourself when you cut yourself. We all have adult stem cells. The problem is we forgot how to really take advantage of these things. Okay? We can't regenerate a new arm, a new finger, right? I mean, I grew up in South Texas, and I remember chasing lizards all the time. You grab their tail, and what happens? Tail comes off. But the lizard doesn't care. Why? Because he can grow that tail back. Okay? But we, as mammals, have forgotten how to take advantage of stem cells, but we're learning how to do this. And we're going to show you that here in a minute. Okay, so let me walk you through embryonic stem cells. This is what we classically think about. This is what you guys have heard in the news. This is where a lot of controversy, admittedly, has been taking place in the field. But this is the embryonic stem cells, kind of the old paradigm of what we used to think about stem cells. And I'm going to show you how we transitioned away from this. Okay, so here's the embryo. The egg is fertilized. And here in the first couple of days, these are those totipotent cells I told you about. They continue to grow and divide. And then you get to what's called a blastocyst. Every single one of you in this room, including myself, were a blastocyst at one time. We just don't remember that we were. It's a long time ago, obviously. These are the cells, the embryonic stem cells, that you can actually take and be able to culture into a petri dish. And then based on various types of additives with these stem cells, you can make these cells differentiate into things like muscle cells. So you can actually use this to potentially damage a heart after a heart attack. You can go after these cells and convince them to generate new neurons. And so this could be something for like Parkinson's disease or Alzheimer's disease. Or you can even convince these cells to convert into pancreatic islet cells. These are the cells that secrete insulin and help us regulate our blood glucose level. And obviously a huge impact with diabetes, which is a growing problem. And I'll walk you through that today. It just so happens that these three things right here on this picture I show you are the exact three things that we're going to talk about today. And I'm going to show you how I can take stem cells and grow heart cells and grow nerve cells and grow pancreatic beta cells. Okay? This is really, really cool stuff. We're having a blast. But I told you, every single one of you, okay, regardless of whether you're 85 or whether you're 16, doesn't make a difference. You have adult stem cells in your body right now, okay? And this is what we actually, in my group, are trying to target. I don't really need embryonic stem cells anymore, and I'm going to show you why we don't need these cells anymore, because we have adult stem cells right now that we can take advantage of. You have an endogenous population of stem cells just waiting for us to figure out how to exploit them and be able to regenerate new tissues in your body. Okay, this is like internal regenerative medicine that we're working on. And again, remember what I'm doing here. I'm a chemist, okay? I got my chemistry degree here. I went to A&M, got a PhD in organic chemistry, and now I'm playing with stem cells, okay? I never dreamed I was going to play with stem cells. In fact, 6 or 7 years ago, guys, I had no idea what a stem cell was. Okay? And this is the great thing about the opportunity you guys have at this day and age, is that all these fields are starting to cross-fertilize. Okay? Chemistry, biology, physics. You have the opportunity to learn all of these disciplines as you go through your career, hopefully here at SFA. And then do grad school at UTSA with me. I had to get that in. Okay? So these are our heroes. Okay? These are the potential cells that we're going to take advantage of and be able to utilize to solve some of our biggest problems in the 21st century. Okay? But there is a dark side to stem cells that I'm going to walk you through as well. And we're going to have to learn what that is. But there's so many unanswered questions. Okay? These stem cells have been around for a very long time. Even before I was even in school, people realized the potential stem cells back in the 70s. Okay? They've been around forever. This is not anything new, but it's taken us this long to figure out how to exploit the use of stem cells, okay? And all these questions we have, especially when it comes to adult stem cells, we have no idea. Are they leftover embryonic stem cells when we all grew up and are still growing? Are these adult stem cells that you still have leftover from when you were an embryo? We don't know. We have no idea, okay? Why do they stay undifferentiated? If you take these cells and put them in a petri dish in a biology lab, especially these embryonic stem cells, and let them sit there, they will sit there and stay a stem cell until they want to, or until you encourage them to differentiate into something else. They just stay there for years. We don't even know how that happens. How can they do that? How can they be immortal? Okay? These are things that we just don't understand. And the problem is we don't understand how they divide, how they proliferate, and how they decide to differentiate. How does that stem cell, when you're growing as an embryo, say, okay, I'm going to be the part of the bone, and I'm going to be part of the skin, I'm going to be part of the brain? We have no idea how that happens, okay? That's really basic biology, right? And we don't even understand how that process takes place. How in the heck are we ever going to utilize these things for therapeutic potential? Okay? Just think about that. It's basic stuff. Scientists today have no idea how an embryo actually develops and divides, and each cell differentiates itself as you grow. That's basic stuff. We have no idea how this is happening, all right? This is something we hope to figure out down the road, but there are a lot of things that we have to address, okay? But I told you we don't really have to worry about embryonic stem cells anymore. Okay, this is really, really cool stuff, guys. Okay, there is now a new way of accessing these embryonic stem cells, or cells that act like embryonic stem cells, that can divide into any cell type you want. And the cool thing is, you know where to get these cells from? You guys. We have the potential to take a skin cell off of your skin or hair or anything like that, and make that cell go backwards, okay? And convince that cell that instead of being a skin cell or a hair cell, we're going to convince you to go backwards in your life cycle and become now an embryonic stem cell. These are called induced pluripotent stem cells, okay? So this is great new technology. A phenomenal scientist in Japan actually figured out how to do this. So here's the old paradigm, right, where we would get these cells, these embryonic stem cells from the growing embryo, and utilize these to take advantage of their capacity to differentiate. But now we can take from this patient and take this patient's skin cells, and with her skin cells, be able to add in factors and various genes to convince these cells to go back to the same type of stem cells that we had from the embryo. This is great technology, okay? This is phenomenal technology, and this is actually something we're doing at UTSA, okay? We can take these cells, okay, and be able to convince them to go backwards into stem cells, and then use those stem cells to regenerate the tissue from that particular patient, okay? Maybe grow some new nerves or some new heart muscle, and put those cells back in that patient, and they will not get rejected. Why? Because these cells came from that patient to begin with. Okay, so we always heard about organ rejection and these kind of things. We now have the technology to go reverse and then go back again and plant and retransplant those cells back into the patient. This is really, really cool stuff. All right? So to give you an example of how stem cells can actually be our heroes in this case, I try to always play this duality of stem cells. They can be heroes or they can be villains. And I have a great movie here to show you. It's really quick, so you got to pay attention really fast. Okay, but this is a study that was actually done by a company in California where they took a mouse, okay, and take this mouse, and they could sever the mouse's spinal cord, and then take cells from that mouse and differentiate those cells back to stem cells, and then convince those stem cells to grow back into neurons, transplant them back into that mouse, and let's see what happens. Okay, so the first part is the mouse after surgery. You see here, he can't move, really. His back limbs are pretty, but this is the same mouse after stem cell transplantation. Look at him now. Okay, yes, this is difficult sometimes for people to see, right? This poor mouse volunteered for these studies, but this is where we have to go, guys. Okay, this is how things move forward. Obviously, we don't want to sever our spinal cords and make sure this works, right? But this is the potential of this technology, okay, to be able to think about it. You know, if there's soldiers out there or other people that have spinal cord injuries, this study right here gives us great encouragement that we can actually solve some of these problems with stem cells, okay? This is a phenomenal opportunity. But there is a huge dark side to stem cells, okay? And this is where things get extremely complicated, guys. This stuff is extremely difficult to manage. And the reason is that this is a picture of a growing tumor inside a patient, a cancer, lung cancer tumor that's growing inside of a patient. And the thing I want you guys to appreciate about cancer, right? There will never be a single cure for cancer. Ever. Ever, ever, ever. Okay? Because every single cancer is different. Lung cancer is different from pancreatic cancer, which is different from breast cancer, which is different from colon cancer. And the problem is that all these cancers between patients, two patients that have lung cancer, those cancers can be very different. This is a horrible disease, okay? A very horrible disease. And the problem is that this growing tumor right here is not just one set of cells. This tumor is a whole mixture of cells, okay? Some cells that help the tumor grow and create blood vessels, other cells are in there to help generate energy. And then the problem is that the core of this tumor, in fact, this is a new paradigm that people are believing in cancer. At the core of this tumor is a cancer stem cell, okay? And this is where the problem becomes. We all, I'm sure, have heard stories. I have stories to tell you about my own family members that have gone through chemotherapy, right, and gone through this horrible treatment of chemotherapy. Looks like the tumor is in remission. What happens 6 months later? Cancer comes back, right? And the reason is that because there's this cancer stem cell at the root of this tumor, our drugs today cannot target those cancer stem cells. These guys are smart. These guys are very smart, okay? And they can take those cancer drugs and pump them right back out, and they get unaffected by this, okay? And to give you a dramatic example of this, this is the tumor here. What they are able to do is to separate the cancer stem cells out of this tumor. They treat those cancer stem cells with some of our best cancer drugs out there in the world. It's supposed to be able to kill this cancer, right? Watch what happens after treating with cancer drug. These are these cancer stem cells. They didn't even care it was there. Look at them divide. They're going crazy, okay? This is why cancers recur. This is why cancers metastasize, in other words, spread. That cancer stem cell will break off in your body and go find a new home and do this all over again, okay? Cancer is tough. Cancer is a really tough disease, and I hate this disease. I hate it. Okay, I've lost 3 grandparents, 2 uncles. I hate this disease. All right? So these are huge problems. And so stem cells have this great potential to regenerate a new organ, a new heart, repair a spinal cord injury. But if we're not careful, we potentially could give our patients cancer. Okay? We have to learn how to control these stem cells, or we're going to give them even a worse disease than what they started with. Okay? This is extremely tough science to be in. All right? So what determines this role? How do you know a stem cell is going to be my hero of the day and solve the problems that I want it to solve? Or how am I going to know that this stem cell is going to divide and give me cancer? What's going to decide this? We don't know. That's the problem. Okay? That's what we got to figure out. Okay? This is going to be tough, guys. This is going to be really tough. Okay? So, I'm going to try to promote to you today that I think stem cells have great capacity to solve some of our biggest health problems today. But before we can do that, before we can even think about going and trying to solve some of these problems, what we have to do first is figure out how they do this. Right? Why does a stem cell divide and generate a new heart muscle cell? Why does it generate a new bone cell? We don't know how these processes even work. Before we can do that, we need to figure out how these stem cells decide what they want to do. Okay? And that's what my group is involved with at UTSA. And the cool thing is that this is where chemistry can help. Okay? Chemistry, as you may already have heard, is the central science, all right? It's the best science, period. I'm biased. Very biased, all right? But it has this great capacity to cross and go into various fields of biology and physics and medicine, which is I'm involved with, to solve some of our biggest problems, okay? And that's the great thing about chemistry. In fact, chemistry today is not just beakers and flasks anymore. You probably know this, right? I'm preaching to the choir. But look, chemistry is the central science because of all these other fields that it can tap into. Medicine and law, education, pharmaceutical, which I was involved with, environmental. There's a lot of cool stuff going on, nanotechnology, and being able to look at individual atoms these days. And the ability to do that is going to help us solve some incredible problems, even in medicine, okay? But chemistry is at the heart of all of this stuff. And that's the great thing and why I'm so happy I got a degree in chemistry, okay? It's one of the most flexible degrees you possibly can have. And that's true about all sciences. You have this capacity to go out in even business and law. A lot of my friends are patent attorneys. They got a degree in chemistry, and now they work in Washington, D.C. for the patent office, and they're patent attorneys. You have a lot of opportunities because you have this great foundation in a hardcore scientific discipline like chemistry, okay? This is why I love what I do. Absolutely love it. All right? So, what I classify myself as is a medicinal chemist. Okay? My job every single day when I go into work every day, I got to teach class and these kind of things, which I just love. Absolutely, because I get to show everybody, all my students, what I did at Merck and show all these great drugs I made. But my job and my research is to be able to translate all these basic discoveries we have, all these compounds that I'm getting ready to show you that we make, all these cool things we do with the stem cells, and translate those basic discoveries at the bench and try to convert them to a new therapy at the bedside. Okay? That's what a medicinal chemist does. It's a great, great opportunity, a great job to work with. Okay? And the cool thing is, as a medicinal chemist, I got to know how to make compounds. I get to study cells and do biochemistry and cell biology, molecular biology. I try to convince a rat to go into an Erlenmeyer flask. Just kidding. I like that picture, though. At least it gives me some perspective. And you get to do cool stuff, okay? And sometimes you even get to blow stuff up, which is even more fun. Not on purpose sometimes, okay? This is actually, this picture I got off of Merck's website. That young woman right there, when I worked at Merck, I actually hired that person. Now she's on the front page of Merck. It's really cool. And I even know what that reaction is. That was a very interesting day when we ran that. Combine all these disciplines as a medicinal chemist: chemistry, organic chemistry, analytical chemistry, biology, biochemistry, cell biology, all these disciplines together that I get to touch and play with to eventually solve and make a new drug. Okay? That's what I do. It's a fun job. Okay? I'm not spending my time, forgive me if there's somebody in here that actually does this, but I'm not trying to make a new plastic for a cool new coffee maker. Right? I'm actually solving some huge problems for our world today, or at least trying to. And the thing that's just hard for me when I wake up every morning is that the compounds I worked on at Merck, like Januvia, this type 2 diabetes drug, I had no idea when I started working on that project that at the end of the day, 2 of my family members, 2 of my aunts would be taking that drug. Okay? So it's easy for me to get up in the day. Okay? I have no problem getting out of bed. I love doing this because the impact that I may have one of these days could be huge. Not only for everybody else in this room, but for everybody else back in McAllen, in Houston, and Dallas, where my family lives. Okay? A great opportunity, guys. Just a phenomenal, phenomenal feeling. All right. Chemistry. You're not going to get away from me without me showing you some chemistry today. All right? I am an organic chemist, and I love to make molecules. And the cool thing is that the power of medicinal chemistry, what I do is to make molecules that actually can convince stem cells to do what I want them to do. Okay? If I could find a molecule to make my 8-year-old daughter do that, that would be great. But I can't do that, apparently. Okay? But there's some great molecules out there that people have already worked on at places like Harvard and Stanford, some of the biggest places and universities in the world, that have already identified molecules that can convince stem cells to do them what they want to do. Okay? Some of these molecules can make new heart cells. Some of these molecules can make new nerve cells. But the problem is they still don't know how these molecules do this. This is tough stuff. Okay? And I want to solve this problem. I need to understand how this works, or I'm never, ever going to be able to translate this to new therapy. Okay? So this is what we do. We make molecules, and we try to figure out how they are actually making stem cells do what they want to do. So here's a couple of different examples of some of the things I've been working on. We'll get into more details, but two of the things that we started off on, and here's our embryonic or our adult stem cells, boring looking blue spots. They're sitting there doing nothing. By simply sprinkling a little bit of compound, a little magic dust, if you will, okay, like this compound that we made in my lab at UT San Antonio, we can convert these stem cells into these heart cells, okay, so we're inducing cardiogenesis, okay, and that's a huge, huge advantage for us, and I'll tell you why in a minute. Or another class of molecules, this compound here can actually induce these exact same stem cells to do neurogenesis, or generate new neurons, okay? This is cool stuff. Again, I get to make these molecules, I get to treat these cells, and I get to look under a microscope and see this cool stuff happening. All right? Every day. It's just a phenomenal opportunity. So, how do we do this? This is where it gets really cool because chemistry has definitely jumped into the 21st century full stream. Okay? This is some of the instrumentation that we're actually building in San Antonio. It's called high throughput screening. What we can do is we can actually screen about 200,000 compounds in a week, okay? And this little plate right here, if you guys see this little plate right here can actually run up to 1,352 experiments at the same time, okay? So every little one of these little wells, these little circles, has a stem cell in it and a compound in it, okay? And we can run these experiments, 1,352 of them at the exact same time. Just think how powerful that is, okay? When I was a student here at SFA, I ran maybe one experiment a day. Now I'm doing 1,352 at the same time. And the cool thing is that we actually use robotics to do this. So let's see if this movie will take off. This is some of the instrumentation. And so these little plates right here are actually what's moving through this instrument. This is all robotic controlled. The robotics introduce the compounds, introduce the cells. All you do is stand back and watch, okay? And let this thing move and do what it wants to do. And it'll keep moving these plates over and over and over again. And that's how we get to actually screen 200,000 compounds in a week. Okay? So this is cool stuff. And I get to learn about robotics, and I get to learn how to control these things and write my own algorithms and some of these things to control exactly what these robots do. This is fun stuff, man. This is really fun stuff. Okay? And you get to interact with this, even as a graduate student, even as an undergraduate student at my institution, UTSA. Okay, and play with some of these things. It's really, really cool stuff. Okay, so and again, you get little colored wells that tell you whether one compound's working or one compound's not. You've got an instrument that'll read this out for you. It spits out the data, and you say, "Oh, that's the compound that's doing what I want it to do." Okay, and then you take that and move it forward. This is how it all starts. So once you do that screen, once you put all that stuff in that robotic system, and it screens all these compounds and tells you this compound is good and this compound is good, you pull out structures, okay? And you know what those compounds are and what those structures are, and then that's when I get involved. Okay? As a medicinal chemist, I look at these compounds like, oh, I like this one, and I like this one, and this one. But you know what? This one over here looks really interesting to me because I know one, that I know I can make it really easily, and there's some other great drugs out there that kind of look like this, and so I want to go after this. And so as a medicinal chemist, we target that structure, and we do medicinal chemistry. In other words, now I'm going to take that lead structure, that scaffold, and I'm going to make a whole bunch of derivatives to try to make that compound more potent, okay, to make it better, to make sure that it's actually going to be a drug someday. Okay, there's a lot of things you have to worry about. We take it for granted that we swallow a pill, right? You take Tylenol, you got a headache. There's a lot of research that went into that particular formulation to make that a pill, right? When my kids take Tylenol because they have a fever and it's a suspension, right? They can't swallow a pill. That suspension, that takes a lot of work just to develop that suspension. Okay, so not only do I have to make a new drug, I have to figure out, is it going to be a capsule? Is it going to be a tablet? Is it going to be a liquid, right? Is it going to be a shot? These are things that we have to deal with all the time, and this is what medicinal chemistry is about. These are the guys that make drugs, and this is what we're trying to do. So we make compounds, and we make a whole bunch of compounds, and then we make more compounds. Okay? This is what my students do. Some of my students do every single day. They come in and they make a whole bunch of compounds. And sometimes they'll make 50 compounds, 60 compounds in a single day. And again, I thought I was doing great when I was in grad school making 2 or 3 compounds a day. Now the technology is out there and combining all of these STEM fields, right? Science and technology, engineering and mathematics, to be able for me to pull off this chemistry in this capacity. Right? It's really, really cool stuff to be able to show how many molecules we can make. But what I want you to just think about these molecules is that they're going to be my keys. All right? There's this huge mystery with stem cell and it's biology. It's a black box. I told you, we don't know how these stem cells work. But my molecules are going to be my keys that are going to hopefully unlock these mysteries of these stem cells. Okay, so I've got these huge set of keys, and sooner or later I'm going to find that key that's going to fit the lock and unlock my stem cell. And once I figure out that, I'll be able to really, really tap into the regenerative capacity of these stem cells. All right? So let's go through 3 stories, 3 little vignettes, if you will, that I'll show you today of some of the things we're working on in my lab at UTSA. All right? And I'm going to talk initially about neurogenesis and brain cancer. Okay? I told you before, every single one of you, right, has active stem cells. Okay? Every single one of us. Okay? And the cool thing is that before we knew we had stem cells in the blood, right? We always heard about bone marrow transplants, right, to help solve some of our diseases of the blood. And those are stem cells that are in your bone marrow. But what we didn't realize is that we actually have stem cells in other areas of the body that we never, ever dreamed possible. And the one that's the most surprising is in the brain. Okay? You have a portion of your brain, area of the brain that's called the hippocampus. Okay, this is where a lot of your short-term memory is stored and long-term memory. Unfortunately, this is one of the first portions of the brain that gets affected by Alzheimer's. Okay? A horrible, horrible disease. Absolutely horrible disease. But the interesting thing is, if you can see these little blue dots right here, okay, those little blue dots are adult stem cells. Your brain is actually making new cells, new neurons right now. Okay? If you drank as much Starbucks coffee as I did, you're probably making more even. You guys are looking. All right, anyways, these blue cells are the ones that are actively dividing and actually making these green cells, which are our active neurons in our body. Okay? And so we want to be able to take advantage of this and be able to tap these stem cells in the hippocampus to be able to go after some of these diseases like Parkinson's and Alzheimer's. All right? So in collaboration with Dr. Jenny Shea, who's actually a professor at UT Southwestern Medical Center up in Dallas, where I used to be, we developed molecules. These are a series of molecules that will actually take these boring stem cells out of the hippocampus. And as you see this pink stuff, this kind of fuchsia color growing in, this is actually a protein that gives us the indication that these cells are differentiating into active neurons. Okay? And these molecules are encouraging these stem cells to do this faster than what they normally would. Okay? And actually better than what they normally would. And so as we develop these molecules and we see that these stem cells are starting to make all these little dendrite formations, so if you look at a nerve cell, right, they've got all these little fingers sticking out of them so they can communicate with each other. That's exactly what this pink stuff is. And we develop new molecules and we can actually track their activity, and we can study a lot of the proteins that are involved with this and understand exactly what's going on. Remember, that's my point. I've got to understand how these stem cells are working. I'm never going to be able to tap into their full potential. And my molecules are now capable of doing this. Okay? So this is what we want to work out as far as how these compounds are working. And the cool thing is that once we figure out which compounds are actually doing what we want them to do, there's these really cool techniques out there. This is a picture of cells. These are live cells. And like this is actually the cells when we just treat them with solution, with no drug, no compound in them. But this lane right here is the lane where we actually put our molecules in. And the color indicates that these cells are releasing calcium. Okay? And the release of calcium is a good indication that these stem cells are starting to differentiate. And you can actually visualize this. You can actually watch this under a microscope as these cells start to release calcium. You can actually image these on this really cool microscope. Okay? And now we understand how our molecules are working. We know they're targeting a specific receptor on the cell, and this causes the release of calcium, and then everything happens downstream from there. Okay? So we're going back to the basic science of this to be able to help translate what we're trying to figure out today. Okay? So here's another cool picture. Here's these boring stem cells, and after we treat them with some of the molecules, this is what these cells turn into. You see these little red things, these little fingers reaching out? That's those cells trying to communicate with the other cells in that petri dish. Okay? These are the stem cells that are actually trying to differentiate. But the real application here that I want to actually be able to show you guys is we will want to apply this back to cancer. Okay? This is a picture of a patient at Parkland Hospital in Dallas that had a very huge brain tumor. Okay? This is glioblastoma multiforme. This is a horrible, horrible cancer. Okay? I've lost 2 uncles to this disease. Okay? And both of them, when they got diagnosed, within a month they were dead. Okay? So you realize that I hate this disease. I hate this disease with a passion. The average lifespan is only 14.6 months, and there's over 22,000 new cases every single year of brain cancer. Okay? The problem is that all our cancer drugs that are out there, they can shrink this tumor, but they never can get rid of it. Why? That cancer stem cell I told you guys about, remember? Okay, this tumor in particular is really, really good at getting rid of cancer drugs, and that cancer stem cell is the reason why. So we took a section of this patient unfortunately died. Okay, we got a section of their tumor. We took that tumor, we isolated the brain cancer stem cells out of this, and these are the cells that are completely resistant to all of our current chemotherapy. We cannot kill these cells. Okay, unless we treat them with really massive amounts of radiation. However, if we take one of the molecules that we made in my lab, we treat these cells, we can convert these cancer stem cells into benign neurons. Okay? Now, this is cool stuff. This is really cool stuff. And so our thought is that we can take existing chemotherapy drugs in combination with some of the things we're making in San Antonio to really go after this disease in a very new way. But guys, we're a very long way from this. Okay? An average time to develop a new drug, and believe me, I did this a lot at Merck, 10 to 15 years to develop a new drug. Over $1 billion to develop a new drug. I don't know about you, but I don't have $1 billion laying around. Okay? We got a long way to go. A long way to go. Okay, but this is really cool stuff. The other thing is that this is actually one of my grad students, Hector Aguilar, who grew up in Corpus Christi, Texas, did his undergraduate degree at UT Pan Am down in the valley, is now a grad student with me. We take these exact same cancer stem cells and we treat them with our drug, okay, or we don't treat them. In this case, we don't treat them with any drug. We can put these cells in a mouse, and that mouse automatically, within a week, starts generating a tumor. Okay, that's these cancer stem cells that can be so aggressive that if you transplant them in a mouse, they will actively start developing a tumor within that mouse. However, if you take these exact same cells and treat them with our drugs that we're making, these cells cannot divide, they do not grow a tumor, they do not cause any problems within that mouse whatsoever. They stay dormant. Okay? So it's a good indication that even in a live animal, some of our compounds are doing what we hope them to do. Okay? Again, guys, we've got a long way to go, a lot of work to do, but this is some real cool stuff, and Hector actually is a phenomenal student. He gets to make the compounds, he gets to work with the stem cells. Again, he gets to translate across all these fields between chemistry and biology. All right, cardiogenesis and heart failure. You guys know there's lots of drugs out there that help you control your cholesterol, right? Statin drugs. But what happens if you have a heart attack? What's your options? What are you going to do if you have a heart attack? Your options are what? Heart transplant? Or you can go on a ventricular device? Bottom line is your life is going to be dramatically altered. And the reason is what happens is that when your heart has a heart attack, your heart loses its blood supply, and that part of the heart gets damaged. And the only way your heart can respond to this is by generating scar tissue. Okay? Because your heart wants to maintain the integrity of its structure. Okay? And the only way it knows how to do that is to generate scar tissue that keeps that integrity in place. But once you have scar tissue, that's not active contracted muscle. Right? And so your heart has lost massive capacity to beat like it normally did. Okay? But if we can convince the heart, instead of making scar tissue, why don't you go ahead and make new heart muscle? That would be a great way to treat patients that have heart attacks, right? There's no really treatments out there for heart attack patients. And that's what we hope to be able to do with some of the things that we're working on in my group. So this is a collaboration with Jay Schneider, who's actually a practicing cardiologist. He treats heart patients at UT Southwestern Medical Center and runs a lab at the same time, okay? This guy is extremely busy. And what he's helping us do is that this is a heart, an image of a heart that's gone under a heart attack, and this whole bottom portion right here is where the scar tissue is formed. We hope to be able to take advantage of the fact that this guy here figured out that your heart, on the outside layer of your heart, there are stem cells. And it's these guys that were making the scar tissue. But they know how to make heart muscle. They just forgot this capacity. Our molecules convince them otherwise. Okay? We can actually convince them now to make heart muscle rather than scar tissue, and then repair the heart, regenerate your heart after a heart attack. Okay? So, this is a picture of a human heart. You know what this picture is? That's a mouse's heart. Okay? And this is the challenge that I have. I have to translate everything that we learned in this little thing here to this organ here. Okay? But the cool thing is that there's a lot of similarities between the mouse heart and the human heart. And the cool thing is right on the outside layer, the epicardium of your heart, this is the sheath around your heart, that's where all these stem cells are at. Okay? And my collaborator figured this out. We know they're there. We just got to take advantage of them. Okay? And those same cells are in the mouse heart as well. So we hope to be able to translate this to the human heart. Okay? So here's a nice image, microscope image of tissue from a normal heart. The green stuff right here is your epicardium. Okay? That's just what it looks like normally. As soon as you have a heart attack, look what happens to the epicardium. Now it's grown dramatically. You have all these green cells that are trying to repair the damage. They're going crazy. They don't know what to do. The only thing they know how to do is do it fast, is to generate scar tissue. That's the only recourse you have when you have a heart attack. Okay? And all these cells in here are trying to do that. But the cool thing is that we can actually now, with our collaborator in Dallas, we can actually take those cells, pull them out of the mouse, isolate them, grow them in a petri dish in the lab, and now play with them in a petri dish and figure out what we can do with them. So the experiment we've designed is we can take the mouse's heart, whoops, go back here a little bit, previous. We can induce a heart attack in these mice. We can take these mice's hearts. We can isolate these cells that are trying to make scar tissue, culture them in a petri dish. Remember that robot I showed you? We can screen all of our compounds that we're making across the world, find a molecule that will convince these cells, instead of making scar tissue, why don't you just make new heart muscle instead? Okay? This is a huge finding for us, a huge, huge finding for us. And so we go back to our keys, back to our molecules, and figure out which ones actually are capable of doing this. And here's a cool picture that shows these are those mouse cells we got from the epicardium. The green stuff, the green stain here, is these cells making collagen, scar tissue. Okay, so if we don't put any drug on them whatsoever, this is what these cells do. However, when we treat them with our compound, our isoxazole, what these compounds are called, all this red stuff, there's these cells generating new cardiomyocytes, new beating heart cells. Okay, so this is really cool. Again, this is in a petri dish, although we have a lot of data in mice as well that allows us to show that we can actually pull this off. And here's a cool picture that shows this. Here's the progenitor cells, the stem cells from the mouse's epicardium of his heart, boring looking cells. If you take these guys and don't do anything with them, they will generate scar tissue, collagen like crazy. You take one of the molecules we developed in my lab and let them sit on there for about 10 days, watch what these cells will do. They start beating, just like we want them to. And that's in a petri dish. Okay? That's not in a live organism, that's in a petri dish. Okay? So this is cool stuff. This is something that we hope to be able to translate this into something that will be valuable one day down the road. Okay? All right, move forward. There we go. One more study. We took mice, and this is something that they do up at Southwestern, 20 mice that didn't get our drug and 20 mice that did get our drug. You can induce a heart attack in these mice. There's 20 mice that didn't get the drug. What happens with these mice, these are the ones that are in black, after 7 days, 50% of these mice die from their heart attack. However, the mice that got our drug before we induced a heart attack, 15 of them, 75%, are still alive. Okay? So this is a real quick experiment to let us know, even though at this point we don't know exactly how these compounds are working, we have a good indication that, at least in mice, again, I've solved a heart attack in mice, great. I still got a lot of work to do down the road, but at least this gives us an indication that there's something, even in a living organism, that we can affect when it comes to heart disease. Okay, and this is another picture of these hearts from these mice. These are the mice that did not get drug treatment, and you can see the damage here. Those mice that survived have a little bit of a heart attack. This is not that much scar tissue, but look at the scar tissue on the mice that did get our drug. These are mice that are still alive. This is massive damage to this mouse's heart. However, our drug is somehow being able to minimize that scar tissue formation internally in this heart and allow these mice to survive these massive heart attacks. Okay? So these are pictures, again, as a chemist, that I would never dream that I would have the ability to show. These are a lot cool pictures than some of the structures I get to show. All right, one last thing I want to talk about, and that's regeneration and diabetes. I can promise you one thing, by the time you guys get done with your education, diabetes will probably be the #1 public enemy out there. Not only in North America, but across the world. And I'm going to show you why this is the case. CDC, the Center of Drug Disease Control, gave some great data. It shows the progression of diabetes in North America. This is data from 2000. The red numbers, the red boxes here show the percent of the population within a given state that has diabetes or has been diagnosed with diabetes. Here's 2000. I'll move forward, please. I think my plug is stuck up here. There we go. 2001, 2002, 2003, 2004. Watch what happens to Texas, guys. 2005, 2006, 2007, 2008, 2009. Okay? And I promise you 2011, 2010, 2011 doesn't look any better. This is huge. Those states in red, over 10% of the population has been diagnosed with diabetes. And the complications of diabetes relate back to cancer, relate back to heart disease. This is something you guys in this room are going to have to tackle. You're going to have to. Okay? We're going to have to figure this out. And the problem is treating diabetes is something that is not easy to control. Okay? Here's one approach for type 1 diabetes. What a potential treatment option is if you can take the pancreatic beta cells, so in your pancreas, those are the cells that generate your insulin. You can take those cells from a cadaver, somebody that has passed away. You can take those cells as soon as that person passes away and be able to take them and transplant them into a person that is developing type 1 diabetes. Will help them manage their glucose levels. Okay? This is one approach. You can take these cadaver cells and transplant them into a patient and let those cells find a home in their pancreas and generate insulin and help control their diabetes. The problem is that ideally you want to take these cells from these cadavers and be able to store them somewhere, right? And as soon as you have a patient that's been diagnosed with diabetes, pull those cells out and be able to transplant them. But the longer you store these cells from these cadavers, the more capacity they lose to secrete insulin. They become diabetic. Those cells in themselves become diabetic. And this creates a huge problem. We would like to have a cell bank to be able to have this. And you can't do this. Okay? So you basically would have to, every time you'd have a patient been diagnosed with diabetes, you would have to have fresh cells from a cadaver to transplant that right within the next couple of days. That's very difficult to do. Okay? Extremely difficult to do. If we could solve this problem, that would be awesome. Well, we have a potential way of going after this now. This is new data. The first time I've really presented this data is that all these molecules I showed you before, guys, these are the same molecules that we showed when the neurogenesis stuff. Well, the same gene that gets turned on in these neurons is the exact same gene that your pancreatic beta cells need to secrete insulin. So we thought, well, heck, we've already got molecules that are inducing this gene or turning on this gene. Why don't we go ahead and treat these molecules on these pancreatic beta cells and see what happens? And lo and behold, once we do that, these are pictures of these pancreatic beta cells. The red stuff is insulin, okay? Just measuring insulin straight up. Once we treat these cells with our compounds, these cells actually secrete insulin almost like a normal beta cell, okay? They don't just release it when they want. They release it only when you treat these cells with glucose, like they're supposed to, okay? Got it? Am I missing something? Huh? Oh, well, you could have came in. That's hilarious. I wonder, I was like, am I missing something? My hair standing up? What am I doing wrong here? All right? All right, but we still have to figure out how this is going to work. All right? There, now everybody's livened up a little bit. All right. So my challenge to you today, before we leave today, what is my challenge? Be a nerd. All right? I'm a nerd. I turned out okay, right? I did okay. Look, and this is a great quote I love as well. All life is an experiment, okay? The more experiments you make, the better. Period. Okay? Great, great quote. The other cool thing, one more thing before we stop. This is my research group at UTSA. Okay? In June, I flew my entire group up to New York to attend a conference. This is actually a barbecue restaurant on 38th Street in Manhattan, Virgil's, and you get to have a little bit of fun as well at the same time. Okay? An awesome opportunity, guys. Thank you very much. Appreciate it.